All right, folks, we've come to the end. We are at the end of our sugar, right? And we are at the end of cellular respiration. And this is the moment we've been waiting for, the electron transport chain. The electron transport chain is by far the most complicated part of this process. And I always like to describe it as like a magic trick, right? If you watch a magician on stage, there's what you see out here, right? There's what you're expected to see. But the real important stuff that's making the trick work is happening someplace else, right? It's happening in the magician's pocket or up the magician's sleeve or someplace like that, right? There's the upfront part, but then there's the real important stuff that's actually happening in the background. And you have to keep that in mind. What's up front is this transport chain, right? This series of molecules, and you don't have to memorize what they are, but these are just embedded molecules that are in the inner membrane of the mitochondria. And they're going to strip, or more to the point, the NADH and FADH2 that we've been generating and collecting are going to be losing their electrons, right, and their hydrogens, and those electrons are just going to go bouncing down this chain, okay? But in the background, we're going to be building up hydrogen, specifically this hydrogen ion, and this is going to force that hydrogen ion to come through the membrane in a process known as chemiosmosis. But we're not going to just let it come through the membrane anywhere. We're going to make it come through this special enzyme called ATP synthase, okay? And this is going to generate mass quantities of ATP. In fact, this is oxidative phosphorylation. So up front, from here up, that's the front part of this process. That's the magic trick, the oohs and ahs. But what's really important, the important part is right here, okay? And notice we're going to finally get to use that oxygen we've been holding on to, right? And you're going to see the rest of that cellular respiration equation, the water, get formed at the end. Okay, so let's get started on it. So this is the front part of the magic trick, right? All the NADHs we've made, right? What did we make? We made 2 in glycolysis, 2 in the link reaction, right? And 6 in the Krebs cycle, right? So we've got a grand total of 10 NADHs plus 2 FADH2s that are all in succession coming in, and they're going to come in and drop off their electrons. Now, the NADHs come in at the very beginning of this process. The FADH2s drop theirs off here at coenzyme Q, or ubiquinone, or however that's pronounced. But essentially what's happening is electrons get dropped off and bouncy down the electron transport chain. And as they bounce, they're going to lose some of their energy. And that's what the magician wants to show the audience, that there it is, right? But that's not really the background, because remember, at the same time that these electrons are being stripped from our electron carriers, remember those electrons were attached to hydrogen, right? So if we look in greater detail, what's happening is, if you're looking here, this is the inner mitochondrial membrane, and this is the inner membrane space, and those electron transport molecules are kind of embedded in this membrane, sort of upstream from this and downstream from what you're seeing here. But as those electrons get dropped off by the NADH and the FADH2, these hydrogen ions start to build up on in this inner membrane space, right? That creates a need for the movement of these hydrogen ions, right? We have to move them through the membrane to try to balance out the concentration on both sides, blah, 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 cells and transport, right? Plus, I mean, having a bunch of hydrogen ion floating around is kind of a dangerous thing, right? We don't like to do that. That creates acidic conditions. So we need these hydrogens to move inside, but we can't just let them get in any way they want to. We've only got these special points that are set aside for the hydrogen to move through, and this is the most specialist of the special points of all. This is the ATP synthase enzyme. This is the master enzyme, the coolest enzyme, the strongest, best, most important enzyme there is, because it provides a space for these little bouncy hydrogens that are bouncing around in this space to come through. That's the chemiosmosis part. Okay. Chemiosmosis is the hydrogens moving through the ATP synthase enzyme. As they come through, this enzyme begins to crank using their energy. Okay. And as the enzyme cranks and cranks and cranks and cranks, it's going to force this part, or it's going to move this part, and the energy generated here is going to take a phosphate and attach it to ADP to generate ATP. So I always think of it, I don't know if this is technically correct, but it helps me to think of it as kind of like a hydroelectric dam, right? If you think of this as like the water and it's moving through this, which is like the dam, you're generating the energy that's needed to drive the synthesis of this ATP. Okay. And because there's a lot of hydrogen moving through, there's a lot of cranking going on and a lot of energy. And that means those ADPs and phosphates can line up and slam together, slam together, slam together. And that's what we call oxidative phosphorylation. What's the oxidative part? We'll get to that. Hold on one sec. Let's fill out what we know on the chart. I think this is a good place to pause. Okay. We know that we are on the inner, mitochondrial, sorry guys, membrane. I keep hitting this, I'm sorry. Okay, so there we are. What's going in? Remember, we had 10 NADH, and they're going to come off as 10 NAD plus, right? And we have 2 FADH2 that are dropping off their electrons, right? And they're going to come off as 2 FAD. Okay, so we're using our hydrogen carriers, our electron carriers here. Okay, so in the process, this is going to set up chemiosmosis, right? We're going to build up these hydrogen ions on the outside of that inner mitochondrial membrane, in the inner mitochondrial membrane space, and then that's going to set up the means for them to move through the ATP synthase molecule. Okay, so far that's what we've got. Okay, so here it is, right? Here's hydrogen, NADH is coming in, dropping off electrons. FADH2 is dropping them off here. The electrons are bouncy, 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 bouncy through the membrane. Okay, just as we expect, right? But the hydrogen ions, as the electrons are passed, right, those hydrogen ions build up outside, and eventually they're going to have to come through an ATP synthase molecule. And you don't see that in the picture, but again, there it is. Okay, so they're going to come through here. So in fact, what we're going to generate here, we're going to bring in lots of ATP, lots of ADP, and we're going to generate 36 to 38 ATP from this. Okay, and this is oxidative phosphorylation. Oxidative phosphorylation. Okay, so oxidative phosphorylation, we're going to get so much ATP from this process. So before, I know 36 to 38 doesn't sound like that much, but if you think about it, really, you had 2 ATP through substrate level, and then 2 from the next big, and think about like glycolysis has 10 steps, right? And you get 2 ATP out of it, right? A net gain of 2. Krebs cycle has all these steps, and then you got to regenerate the original molecule so you can go back and start the cycle over again. You get 2 ATP out of the process. Big deal, right? And then all of a sudden you get to this step, and now we can take all these components that we've been generating all along, all these electron carriers, we can use them and their energy, the electrons and the hydrogens through chemiosmosis and oxidative phosphorylation will generate a whole bunch of ATP for us. Now, here's the other thing, though, that happens. Again, hydrogen ions bouncing around. We can't have that inside of the cell. So we moved our problem of the bouncing hydrogen ions from the inner membrane space of the mitochondria. Now we've moved it back into the matrix. Yay, right? Good for us. We still have a problem, right? No, we don't, because at the end of this process sits oxygen gas. And oxygen gas, with open arms, it accepts our little bouncy hydrogens after they come in. They've done their job for us. They've helped us to generate ATP. Now oxygen says, come on, come on. And oxygen will adopt, each half of an oxygen molecule will adopt 2 of those little bouncy hydrogen ions. And then at the end of the electron transport chain, the electrons that have been bouncing around will also be accepted by that oxygen, which is why we say that oxygen is the final electron acceptor, right? And it will be used to generate water. Okay. So again, the sleight of hand here is the buildup of hydrogen, the movement of that hydrogen through the ATP synthase, which is going to generate our ATP. That's the magic trick. Yay, the ATP is there all of a sudden, right? But then at the same time, then we're also going to make the situation safe by accepting the electrons and the hydrogens by oxygen to generate water. And that's really important, right? So let's go back and fill out the rest of this. So O2 is coming in, and it's going to come out as water, right? And oxygen is that final electron acceptor there, okay? And that's what's important to know about the electron transport chain. Now, we're not quite done yet. There are a few things that we do need to do to wrap up cellular respiration, okay? One of the things that you have to understand is that evolution doesn't create new, it builds upon what already exists, right? So when these metabolic processes evolved in organisms, you can kind of think of it as anaerobic respiration was probably first because the atmosphere didn't contain free oxygen, so there was no way to do aerobic respiration. And then aerobic respiration kind of evolved. You started with glycolysis, and then new reactions kind of got tacked onto that to create this aerobic respiration as we know it today. But also, these are the backbone processes that end up evolving new means of breaking down and also creating new molecules. So there's not a separate way. I know we started with glucose here, but pretty much cellular respiration is the way that most of your chemicals get broken down, that your energy molecules get broken down, and energy generated from them. So it's not just sugar. Glucose enters right at the beginning and it goes all the way through. But you can actually metabolize proteins because once they're broken down into the amino acids can go into the process separately. Glycerol and fatty acids can be broken apart and go into these reactions as well. And so this same set of reactions, right, has evolved to be able to not only metabolize carbohydrates, specifically glucose, which will go through all of these steps, but notice amino acids can enter glycolysis, they can be converted to pyruvate. Actually, they don't enter glycolysis. They are converted to pyruvate and can continue on. Some of them can be converted to acetyl-CoA, and some of them can even be converted to molecules that can attach to the intermediates in the Krebs cycle. Okay. You do have to take that amino group off of the end of it, though. Again, fats can be broken down to fatty acids and glycerol. Glycerol can go through glycolysis. The fatty acids can be processed into acetyl-CoA and then keep moving. Okay. So we didn't evolve separate processes to digest each of those components that we can actually use for energy. We have the same process. It's just we modified the molecules a little differently to get them through the process. The other thing is, as you might have guessed, some of the intermediate molecules that we create can actually be used for anabolism. In other words, we can actually siphon off some of the products of cellular respiration, some of the intermediates that we create, and use those to make other useful chemicals. Again, evolution doesn't create new processes. It builds on the processes that already exist, right? So over time, metabolism has evolved to be able to use some of the stuff we're producing through cellular respiration anyway and create other stuff with it. So you can actually siphon off of molecules from the Krebs cycle to make amino acids that you need, right? One thing, we didn't mention this in the biochemistry unit, some of the amino acids, there's 20 amino acids, right? 8 of those amino acids, actually, there's more than that could theoretically exist, but there are 20 used in living things. But 8 of those 20 are what we call essential. And when a nutritionist says essential, what that means is the body can't make them through cellular respiration. And the only way that you can get those is by eating them in your diet. Okay. Just as a little side note, point of interest there. But the rest of the amino acids, the other 12, you can actually make using these processes. So now you know when they say essential vitamins and minerals, what they mean, that to a nutritionist, the term essential means you have to get it in your diet if you're going to get it. All right, so this diagram is showing you the rest of this, right? This is showing you now, instead of things going in, it's showing you what you can pull out, right? So you can actually pull glucose and you can tear it apart if you can siphon some of it away, and you can synthesize nucleotides for DNA and RNA that way. Glucose also can be strung together to make starches, right? Like glycogen, right? Pyruvate through fermentation can make lactate, which can be reconverted into glucose. Okay, if it's necessary. You can pull these acetyl-CoAs off and create new fatty acids, right? If you need fatty acids for your phospholipids in your membranes, you need a way to make them, right? And so that's one way you can make them. If you need fats for, say, lubricating joints or, like your skin, so your skin doesn't dry out or your hair doesn't dry out, that's how you can make those fats as well. And then a bunch of the intermediates from the Krebs cycle can be used to create amino acids. And that's what that is showing you as well. Finally, there is a certain amount of feedback regulation that can occur with cellular respiration. When you create or you use up ATP, the cell is triggered to catabolize fats and carbohydrates that are stored, so that more ATP can be generated in your cells. There's a triggering mechanism to kind of kick up cellular respiration when your ATP level drops, and to say to your body, hey, we don't have enough glucose here. Let's start metabolizing some of that starch we've got stored, or let's go and find some of those fatty deposits and start generating glucose for us. Also, there's an enzyme in glucose called phosphofructokinase that's actually in glycolysis that's inhibited by ATP. So when you get a buildup of ATP in the cell, you can actually get negative feedback that turns off cellular respiration at glycolysis, because this enzyme doesn't work, and therefore it's not going to produce the intermediate that needs to go on to the next step. But phosphofructokinase is actually stimulated by AMP, which is adenosine monophosphate. So you can actually further break down ATP into ADP and then take another phosphate off to create AMP. And as you run out of ATP and you start to accumulate AMP, that's going to stimulate that enzyme. Again, that enzyme is also sensitive to citrate from the Krebs cycle. So if you've got a huge buildup of citric acid or citrate, what that means is you're feeding stuff into the Krebs cycle quite a bit, right? Because that acetyl-CoA is binding to it, and that's going to keep glycolysis rolling. If you don't have a lot of citric acid, glycolysis can kick up to feed more pyruvate and then ultimately acetyl-CoA into the Krebs cycle. When you have an excess of that citrate, then that might be a symbol that you need to slow down for a bit, right? You're just kind of trying to synchronize those two processes so you don't overwhelm the molecules of the electron transport chain. So regulation by feedback. And again, that's just reminding you that citrate is what the acetyl-CoA binds to, right? And that step also is regulated by ATP, okay? And again, if you have too much ATP, you can actually stop the Krebs cycle at any of these processes so that you don't get a buildup of ATP.